Chapter 6, Video 2. Today we're going to talk about actually performing the puncture, the dermal puncture, okay? So when we talk about the actual performing the dermal puncture, there's certain techniques that you must use and be familiar with in order for this process to go smoothly and for you to retrieve accurate results. First, we're going to talk about some technical tips. Failure to place the device firmly on the skin is the primary cause of insufficient blood flow, okay? So when we're performing the dermal puncture, we need to make sure that that device is pressed up against the skin firmly, because that will make sure that the lancet actually penetrates the skin like it should. A firm puncture is actually less painful for the adult than too many punctures. So you would rather get it right the first time instead of halfway poking the skin and not getting enough blood flow and having to do it again. So a firm puncture is actually less painful. We want to make sure that we puncture across the fingertip to keep the blood from running. So you're going to look at the skin and you're going to look at the lines and the way the skin lays, and you want to go across the grain. That will keep that blood from running and it'll help it bleb up. Another thing, hemolysis is seen more frequently in dermal punctures versus venipunctures because you may excessively squeeze the site. So we don't want to excessively squeeze that site because we could mix the other body fluids and the interstitial fluids in with the blood. So just know that hemolysis is seen more frequently with dermal puncture because of this reason than it is with venipuncture. Another thing that we need to take into consideration is newborns have an increased red blood cell count. So the red blood cells in newborns tend to be more fragile. So with sample collection, this is the steps that you should take with sample collection of dermal puncture. Always remember, always, step 1 is first drop is wiped off, okay? So we're going to clean the skin with alcohol, let it dry, then we're going to wipe that first bleb of blood off because it may be mixed with alcohol and it may be mixed with tissue fluid. So that is the reason that the first drop is wiped off. After alternate pressure and relaxation, we're going to apply alternate pressure and relaxation. We don't want to scrape or touch the puncture site with the collection device. And then, of course, the capillary tube collection, we want to make sure there's no air bubbles, so we're going to hold it horizontally. And then we're going to fill that container. We're going to put it up there on a slant and fill that container. No more than 2 attempts can be, if you're performing dermal puncture, venipuncture, or anything, no more than 2 attempts. If you can't get it within 2 attempts, then we need to get another blood collector in there because we don't want to torture that patient. Applying pressure about a ½ inch away from the puncture site frequently produces better blood flow than does pressure very close. So remember that. We want to keep our pressure away, and that helps push that blood up. Rotating the hand 90 degrees allows the blood collector to clearly see the drop of blood without placing themselves in an awkward position. And then we have an order of collection. You're going to see this on the test. This is order of collection for the dermal puncture, okay? Capillary blood glasses first, blood smears, EDTA tubes, other anticoagulated tubes, serum tubes, and then we're going to gently mix the anticoagulated tubes for 5 to 10 inversions. We want to make sure that there's no bubbles in here because that could skew blood gases. The order of draw is so important when we think about platelets adhering to the puncture site. So that's why if this puncture site starts to heal and these platelets start adhering to it, that is one reason why order of draw is so important here. We will apply direct pressure. We may elevate the arm or the site. I'm sorry, guys. We may elevate the site. We want to also check and make sure bleeding has stopped when we perform dermal puncture. No bandage for children under 2 years of age. This is a choking hazard, okay? The child may be they may be sensitive to glue, and the child may even eat the bandage and become choked because they're so young. So we do not bandage children under 2. Also, we need to know the information that's required to go on these blood samples. So that's something that I want you guys to look back at. Know what information and know how to label the samples. Labeling the venipuncture samples is equally important. We need to label the dermal puncture samples as well. So the same information is required that is required for venipuncture. We may have to have the microcollection tube extenders and a large pipette. So just making sure that you put all the correct information on this is important. And then we're going to dispose of the supplies in the same way. We may complete a log sheet. And, again, documentation of everything. So I'm going to scroll down. We're just going to go through some pictures. This is an example of the supplies. This is them getting ready to stick. This is them placing the lancet up against the skin. We're wiping away that first drop of blood. Now we've got enough blood. We're putting this blood into the evacuated tube system. Then we're giving them back, holding pressure, and then we're labeling it. We always check 2 patient identifiers. That child can't talk to you, but he does have an armband. He does have a band to verify with. So you ask the parents or get a second person to verify with you that is the correct patient. Remember, with bilirubin, light will destroy the bilirubin, so we need to protect it from light. We need to avoid hemolysis. Detection here is very critical. This is a special collection kit for newborns. This shows you how much blood is acceptable and not acceptable. This is acceptable over here. This is acceptable. This is uneven and this is not filled. So it's not enough blood. If you have any questions, please let me know.